Hello and welcome to today's seminar. My name is Angeli and I'll be producing today's event. Before we begin, just a few housekeeping items. I'd like to let you know this session is being recorded for you to review and for others that could not attend today's live session. We'll be answering your questions at the end of the presentation today. We encourage you to type and submit your questions during the presentation. To do so, click on the Q&A tab on the bottom left of your webinar window. Click the submit button to send in your question. To download a copy of today's presentations, navigate to the materials tab. And if you need assistance at any time, click on the Help tab for our support team's contact information. Great, I think we're ready to begin. Today's Antibody Society's web event is titled Antibodies to Watch in 2021. Our speakers today are Dr. Janice Reichert and Nick Hutchinson. Jan, if you're ready, the floor is all yours.
All right, well, thanks so much. And thanks everybody for joining the webinar. A good day to you. As was mentioned, I'm Jan Reichert, Executive Director of the Antibody Society and Editor-in-Chief of the journal mAbs. I'm joined today by Nick Hutchinson, who is the Business Steering Group Leader for Mammalian Cell Culture at Fujifilm. I'm very pleased to announce to you that the 12th annual Antibodies to Watch paper has been accepted for publication, and I'm happy to update you on the contents today. Following my presentation, Dr. Hutchinson will discuss how the pandemic has affected production of antibody therapeutics. So I'll start off today's presentation with discussion of the anti-SARS-CoV-2 antibody therapeutics, which absolutely qualify as antibodies to watch now and into 2021. We have information on over 200 biotechnology-derived targeted therapies for COVID-19, but due to the volume of that data and the limited time, I'm going to focus today on only the molecules that target the virus itself. I'll discuss the late-stage clinical studies and emergency use authorizations for these molecules. I'll then update you on the usual antibodies to watch content, which includes the molecules that were approved so far this year in the US or EU, and those in regulatory review. I'll finish with a brief mention of marketing application submissions that may occur by the end of 2021. So let's move forward, starting with the anti-SARS-CoV-2 antibody therapies. As part of a collaboration with the Chinese Antibody Society, we're tracking over 120 commercially sponsored molecules or programs where the goal is to produce an anti-SARS-CoV-2 biologic. These are all sponsored by commercial firms. Most of these molecules are monoclonal antibodies. Of the mAbs, most of them are full-length monospecific molecules, but a few are nanobodies or an alternative format such as a bispecific. About 12% are a biologic of some other type of matter. So it could be a DARPin, for example, designed anchor and repeat protein, an Fc fusion protein, could also be nucleic acid that encodes an antibody. We've also included some recombinant or transgenic derived molecules. So these are polyclonal, though. So we do have a few of those in the mixture. To the best of our knowledge, the SARS-CoV-2 spike protein is the target for all of these molecules, and usually the epitope is within the receptor binding domain of that spike protein. This graph shows the most advanced phase of development for the molecules. Not surprising, considering the very short period of time people have had to work on this particular problem. The programs, for the most part, are still in the discovery or preclinical stage. Most of the molecules that have moved forward, you see here, there's just a total of 19 of them. We have 9 currently showing as in phase 1 studies, 4 that transition to phase 2, an additional 4 that are in late stage clinical studies, and remarkably, given, again, it's only been about 10 months or so since people had a chance even to attack this problem, 2 of them have already received emergency use authorizations from the FDA. So I'll provide details about the authorized products and the molecules in late stage clinical studies If you want information about the other molecules, those in early stage clinical studies or those in discovery preclinical space, I invite you to go to the Society's COVID-19 Biologics Tracker page. You'll find a table of the antibodies that are in clinical studies, and you'll find a link to an online searchable database that's on the website of the Chinese Antibody Society. So please do have a look at that when you get a chance. Right now, I'm going to discuss the antibodies to watch, where they are the anti-SARS-CoV-2 antibodies. So these are monotherapies, so they're single antibodies or mixtures, and they're sponsored by Celltech, AstraZeneca, Celltrion, Vir Biotechnology with their partner GlaxoSmithKline, Eli Lilly, as well as Regeneron. Now, please note that these companies may also be working on other molecules or combinations that are in early phases of clinical development. So here I'm just focusing on the most advanced drugs. The most recent entrant into the table is Cell's CSCTA01. A placebo-controlled phase 2-3 clinical study will evaluate safety and efficacy of this molecule in hospitalized patients with severe COVID-19. That's due to start in February and it has a primary completion date in May of 2021. So that's let me just emphasize how fast that is. It typically takes years to get through late-stage clinical studies, and here we are doing it in the space of months. As I discuss the other molecules, I want to draw to your attention that it's very important to understand the patient populations that I'll be talking about. There are three. There are pre-exposure patients. So these are patients who are negative for the virus, and this is a prophylactic use of the drug. There are patients that test positive, but they're in early stages of the infection. They may only have no or mild symptoms of the disease. Then you have the hospitalized patients with severe COVID-19. And I'd like you to pay attention to which patient population I'm talking about for each of these drugs, because that has a clear effect on the emergency use authorization. The drug will only be authorized for a very specific patient population. So the AstraZeneca's mixture, AZD7442, is a cocktail of two antibodies. They were both derived from convalescent patients. The antibodies were discovered at Vanderbilt University Medical Center and licensed to AstraZeneca. AstraZeneca did engineering on the molecule. They do have half-life extension and reduced FC receptor binding. The phase 3 studies are listed, last that I looked, which was just yesterday, as not yet recruiting on clinicaltrials.gov, but they should be starting very, very soon at this point. The phase 3 studies will evaluate AZD7442 in both pre-exposure as well as post-exposure use. So again, AZD7442 is a mixture of two antibodies. Celltrion's is a monotherapy, so CTP59 is a single antibody. It's being evaluated in a placebo-controlled phase 2-3 study. That study is including outpatients, so these are ambulatory people who have tested positive, but they only have mild or no symptoms. That study started in late September, but has a primary completion date already in December, again emphasizing just how quickly these clinical studies are being run. Vir 7831 is being evaluated as early treatment for COVID-19 in patients who are at high risk of hospitalizations. So this phase 2-3 study, again, here we have a primary completion date that's coming up relatively soon. The last two products, which hopefully you know about, Lilly and Regeneron's molecules or products, they both have EUAs. Lilly's, so this is LYCO 555, it received emergency use authorization on November 9th. For patients with mild to moderate COVID-19, its use in hospitalized patients is specifically excluded. So this is a monotherapy. It's also being evaluated for how well it prevents SARS-CoV-2 infection and COVID-19 in residents and staff in nursing homes and assisted living facilities. So there's an additional phase 3. This active trial is comparing the effects of the monotherapy of the antibody versus remdesivir. So in hospitalized patients, remdesivir does have an approval, but its use is only in hospitalized patients. So it'll be interesting to see the results of that study because it is head-to-head the antibody versus remdesivir. Last but not least, we have Regeneron's mixture. This is a 2-antibody cocktail, REGN-CoV-2. It's received emergency use authorization relatively recently, just on the 20th of November. That EUA was also for mild to moderate COVID-19. It's also in late-stage clinical studies to test the effect on hospitalized patients, but also in a pre-exposure setting, so whether it prevents infection in household contacts of people who are already infected with the virus. So a lot going on with these molecules in late stage studies, and still a lot going on with clinical studies of the ones that already have emergency use authorization. So I just want to mention a few other things about these EUAs. Lilly has stated they have 100,000 doses ready to go and may have up to 1 million by the end of the year. So that's really good to have these available for anybody who needs them. Recommended dose is 700 mgs. This is infused over the course of an hour. So that's what's expected of patients. They do have to hold still for an hour while this stuff is infused. Lilly anticipates submission of another EUA for a combination therapy that would include LY-CoV555 as well as another anti-SARS-CoV-2 antibody. And they may have data to support a BLA submission as early as Q2 of 2021. So they are looking to get a full approval for that combination. The Regeneron's, as you know by now, is a mixture of the 2 antibodies. Their low dose was already 2.4 grams of the combination. And it is also for mild to moderate COVID-19, again, specifically excluded for hospitalized patients, but it is for patients who are at high risk for poor outcomes. They do have a contract with the US government to manufacture and supply the material. Some of those doses will thus be available to Americans for free. The authorized dose is 1.2 grams of each of the antibodies, and like the Lilly molecule, it's infused over the course of 1 hour. Just want to mention about the price. Lilly did explicitly state a price is $1,250 for high-income countries. It's a government price. The government price for middle-income and low-income countries would be less. Regeneron, I don't believe they explicitly stated a price, but if you just do the calculation, the 450 million divided by 300,000 doses, you get $1,500 per the vial, which would be the full dose. I just want to point out that despite the fact that antibodies are considered high-cost drugs, the cost for these 2 antibody products is actually less, somewhat substantially less, I would say, than remdesivir. And because there are so many other antibodies in the pipeline, one would expect that the antibodies coming online, getting additional EUAs in the future, would undercut these prices. So we do expect prices to come down with the competition from other anti-SARS-CoV-2 targeted therapeutics. So with that little summary of what's going on in the anti-SARS-CoV-2 space, I'll now move on to the traditional content for antibodies to watch and cover US and EU approvals in 2020. This graph shows the number of antibody therapeutics approved in the 2 regions going back to 1997. That was the first year in which more than 1 antibody was approved. It's obvious that the biopharmaceutical industry has gotten quite good at antibody therapeutics development in the past decade or so. 6 or more approvals have been granted each year since 2014, whereas that threshold was reached in only 1 year prior to 2014. I'm pleased to say that despite the pandemic, FDA seems to have kept the review process on track. So far, 10 have been approved in 2020, but there's still time for 2 more approvals based on the FDA's PDUFA dates. The action, so that's an action date. I want to emphasize that the action does not have to be an approval, but I remain eternally optimistic and hope that these will be approvals that we see in December for 2 more antibodies, making a total of 12 for the year. 2021 also looks like it will be a good year for approvals, and I'll tell you why in a few moments. All right, so let's go over the approvals that occurred in 2020. And these are approvals, all of them are approvals in the US, with only a couple of them approved so far in the EU. So starting out the year, we had teprotumumab, which was approved for thyroid eye disease. We had an antibody approved for migraine prevention that targets calcitonin G-related peptide. Isatuximab was approved for multiple myeloma. It's also approved in the European Union. We had an ADC that targets TROP2 that was approved for triple-negative breast cancer, and also an anti-CD19 antibody for treatment of neuromyelitis optica spectrum disorder, and that was in June. Moving on to the second half of the year, tafasitamab was approved for diffuse large B-cell lymphoma. Another ADC, this one targeting B-cell maturation antigen, was approved for multiple myeloma. Satralizumab became the second antibody approved for neuromyelitis optica spectrum disorder. Regeneron did receive an approval this year for their Ebola virus, their antibody mixture for Ebola virus infection that was approved by FDA in October. The most recent approval was for naxitamab, that's for neuroblastoma, and that was approved just on November 25th. So following that quick summary of what has already been approved this year, I'll now discuss what might happen in the future. Amazingly, despite the pandemic and the fact that 10 were already approved, there are 17, not 16. I found out about another one just yesterday. 17 antibody therapeutics that are currently undergoing regulatory review in either the US or the EU. The 2 antibodies that I think might again qualify this, might be approved in December are margetuximab for breast cancer and tanezumab for osteoarthritis pain. They both have an opportunity, let's say, for approval in December, but we have to wait to see what happens. Tanezumab is also undergoing review in the European Union. So now I'm going to go through the remaining 15 antibodies that are more likely to be approved in 2021. Don't actually have PDUFA dates for these 2, but narsoplimab is an IgG4 antibody targeting MASP2 for hematopoietic stem cell transplant-associated thrombotic microangiopathy. Dostarlimab is an anti-PD-1 for recurrent microsatellite instability high tumors. Dostarlimab is undergoing review in both the US and the EU. Now I'll go over these 5 which have PDUFA dates that I know about that run through February to September. With one notable exception, I would hope there certainly is opportunity here for approval in 2021. So 4 of the 5 shown here are also, they're undergoing review in both the US and the EU. So the antibodies currently in review include an anti-angiopoietin-like protein 3 antibody for hypercholesterolemia. We have an ADC targeting CD19 for diffuse large B-cell lymphoma. Tralokinumab is for atopic dermatitis. It targets interleukin-13. And anifrolumab is for lupus. That one is undergoing review in both the US and the EU, and that has the target date in September. The one I've skipped over, is Biogen's anti-amyloid beta antibody for early Alzheimer's disease. Excuse me. FDA did have an advisory committee that had a recent meeting at which most of the members of the committee voted that the study data did not support the drug's efficacy. FDA does typically follow the advice of their advisory committee, which was negative in this case. There have been exceptions in the past, so we'll just have to wait to see what FDA does in terms of their response, which is due on or about March 7th. All right, there are more possible exceptions or more possible approvals in 2021. I hope there aren't any more exceptions to my expectations that these will actually be approved. Teplizumab may be approved for type 1 diabetes. Inelimumab, again, that is a mAbs, so that is a murine antibody targeting CD25 for acute graft versus host disease. There is another Ebola virus antibody, targeting Ebola virus that is for Ebola infection, undergoing US review. Bimekizumab is an anti-IL-17A antibody for psoriasis that's undergoing both US as well as EU review. Regarding sutimlimab, you may have noticed this did not get an approval on its PDUFA date. It is for cold agglutinin disease. It still is under US review though. FDA issued a complete response letter, and they did cite some deficiencies that related to the manufacturing of the product. So Sanofi is working to resolve issues. The BLA is considered under review because the reviews come in cycles. So BLA submitted, FDA has a review period. At the end of that period, if it's not an approval, we roll into what's called sponsor response time. So right now we are in the sponsor response time. Assuming the deficiencies are adequately addressed, then we'll go into another FDA review period. There is still time for a 2021 approval for that molecule, but of course, we'll have to wait to see what happens. So closing out the set of antibodies that are in regulatory review, we have an immunotoxin for bladder cancer that's in US review. And we also have an antibody targeting PD-1 that is for cervical cancer. And that one is also in US review. Not shown here is the one that I found out about yesterday. Ublituximab is an anti-CD20 antibody for chronic lymphocytic leukemia. The sponsoring company, TG Therapeutics, announced, it was on December 1, I noticed it on December 2, that a rolling BLA had been started. So that one is officially in the list of antibodies that are in regulatory review. But again, I want to emphasize, we have to wait for the PDUFA date to see what will happen. I also want to emphasize that I may not have everything. It is possible for other things to be approved in 21, which can happen in the US if a BLA is submitted before the end of June 2021, if that BLA receives a priority review, and if the first action is an approval. I mean, I realize that is an awful lot of ifs, but potentially, in theory, it could happen. And I want to mention also that I do maintain a list of the US and EU approved antibodies right on the website of the Antibody Society. So if you're interested in that, the list also includes all the antibodies that are in regulatory review. I do update that on a regular basis, so it's a good source of information for what is in the queue for approval. Now, typically I would spend more time on the antibody therapeutics for which BLAs or MAAs may be submitted in the next 13 months or so. This year, the anti-SARS-CoV-2 antibodies just took a much higher priority. Briefly, though, I can say that there should be no problem with replenishing the queue of antibodies in regulatory review as the approvals occur. Of the antibodies that are in late-stage studies, marketing applications for at least 18 may be submitted. 6 of them are for non-cancer indication. It's actually 12 for cancer indications because you have to delete ublituximab now. This happens to me all the time. I put together a presentation and then a day later something else happens. So please excuse that little bobble there. Ublituximab should not be on the list anymore. Now, since the Antibodies to Watch in 2021 paper has been accepted for publication in mAbs, and because you so nicely showed up today for this presentation, I have gone ahead just this morning and posted the accepted manuscript version of the paper on the Antibody Society's website. So if you're interested in seeing this paper, which I hope you got some time because it is 60 pages long, and it's a little over 20,000 words. It's enormous, but it's posted right on the Antibody Society's website in the publications section. So I hope you take a moment to download it, and hope you appreciate the effort that went into that one. In my last graph here, I just want to remark that really that late stage pipeline that I track so carefully and document so carefully in Antibodies to Watch papers, that's really just a tiny portion of the overall pipeline. If you look back at phase 2, you see there's nearly 300 antibodies that have advanced to phase 2 and no further. And if you step back again, you see that around 450 antibodies are at phase 1 and have not yet advanced any further. So there are a very large number of antibodies that are in early stages of development, and that really has helped to keep the number in phase 3 or in late stage clinical studies pretty high, making it a challenge for me every year to document. But that really is what's driving all of these antibodies flowing into regulatory review and onto approval. So well done, everyone. Good job at getting these things into the clinic and through the process. Now I'll just wrap up with some key messages. So take home these messages, which is pandemic is going to continue well into 2021. That shouldn't be news to anybody. We're definitely going to need lots of effective therapeutics and vaccines. There has been a really extraordinary response by so many organizations located across the globe in developing anti-SARS-CoV-2 biologics. It's quite possible, if you include all countries, it's quite possible that some 15 or 20 of these could eventually get emergency use authorizations, of course, assuming safety and efficacy is demonstrated in their late-stage clinical studies. My projections indicate 2020 as well as 2021 may be at or near record levels for the numbers of approvals in the US or the EU, despite the very tremendous demands being put on the regulatory agencies by the pandemic. And finally, do watch for Antibodies to Watch in 2021. The properly formatted version of it should be posted on the mAbs website, hopefully late December, at the very, very latest, early January 2021. With that, I do want to thank my co-author for her assistance on Antibodies to Watch on 2021. It was a massive effort, and I could not have done it without her assistance. I also want to thank the many volunteers from the Chinese Antibody Society for their assistance in collecting all of this data related to the COVID-19 biologics. Finally, I'd also like to thank all of the corporate sponsors of the Antibody Society for their support. The logos are shown here in alphabetical order, and if you see your company's logo in here and you're not a member of the society already, please do go to the website and register for your free membership. Your company has already paid for it, so please do register because you get some benefits for it. Lastly, thank you for your kind attention. We will take questions at the end, so please do put some questions in the Q&A box when you have an opportunity. And with that, I will turn you over to our next speaker, Nick Hutchinson.
Thank you very much, Jan. Thank you for an excellent presentation. the introduction, and hello to everyone that's on the call today. What I wanted to do really was talk a little bit about the data that Jan's shown, which shows that there's really very robust pipelines of antibodies, even a pandemic struck and companies tried to get their anti-SARS-CoV-2 antibodies into the clinic. And those anti-SARS-CoV-2 antibodies have a couple of characteristics about the development programs. Firstly, the great need for speed of development in order to get them into the clinic and then commercialized as quickly as possible. And also, as Jan points out, the fact that they're needed for a very large patient population, but also in relatively high doses, which means that they need to be manufactured in quite significant quantities. And from my point of view, I think that in some ways that the industry was already kind of wrestling with those kind of challenges already. So even if you're not in a pandemic situation, speed of development was important or is important to companies developing antibody therapeutics. And whereas kind of meeting large-scale commercial demand is perhaps less of a focus for the industry, it's without it being a completely unheard of challenge. It links to another challenge the industry has traditionally faced, which is the timing of investments into large-scale facilities and making those large investments many years before the facilities are actually required to be online, when there's quite a lot of uncertainty around how successful products are likely to be. So what I wanted to do in a few slides after Jan's presentation was really just talk to you about how companies are addressing these challenges during the pandemic, what we might learn from the development in the pandemic, and then how we can apply this to antibodies in the future. So starting with the question of timelines first, this is a slide which taken with an image taken from a publication by Brian Kelly. I think Brian might actually be in the audience today, so hi Brian. It's a fantastic paper. Really, what it shows in this image is how the timeline for a traditional program from candidate selection through to IND has traditionally taken around 12 months for the kind of the best-in-class programs. But what Brian set out to do in his paper, and the reference is given at the bottom of this slide, is he proposed a number of adjustments that could be made to programs so that an accelerated program could actually reach IND in almost half the time, so in as little as 6 months. So really fantastic and worth reading. So I urge you to go and look that up. I didn't want to necessarily repeat each element of the paper, but at a high level, I think one of the things that's very important in meeting those rapid timelines, especially for antibodies, is the use of platform processes. And these need to be robust and scalable. And this allows, allows scientists and engineers to really focus not on performing full process development, but really just fitting their candidate to the manufacturing platform. And this can save a lot of time. I guess the downside is that it's not as fully optimized as it possibly could be, but the productivity of modern cell lines and processes is such that if you scale up to a reasonable scale for your first in human clinical trials, you're likely to end up with more than enough material for those studies. And then you can shift optimization to be performed during the phase 1, phase 2 clinical trials when this work comes off the critical path to commercialization. I think some of the other things to call out that we see companies who are looking to develop antibody products very quickly are whereas traditionally people have tended to use clonal cell lines and material generated from clonal cell lines for process and formulation development, we see the increased use of transfectant pools and pools of clones for both formulation development, for the generation of material for tox studies, and even for GMP material. We ourselves use material from transfectant pools to begin formulation development for clients. And I think, the companies such as BMS and Genentech have reported on the use of pools of clones for tox studies. And recently, Sorrento Therapeutics, when they published information about the development of their anti-SARS-CoV-2 antibody, they reported on the use of transfectant pools. I guess that there's some risk associated with this in terms of perhaps not getting, it's possible you wouldn't get comparability, and this could lead to some delays or rework in the form of bridging studies. But in the main, it's an approach that is being used quite a lot these days. Formulation development is something else that's typically on the critical path. We see companies increasingly using, again, kind of a platform approach, so historical data that they have around what formulations will work for antibodies. Sometimes they'll use a lower product concentration for these formulation studies than they intend to use for the commercial product. And again, the risk may be that for occasionally when using this approach, or on some programs, it may be that this approach leads to poor stability, rather than if you're using a fully optimized formulation for phase 1. And this could delay regulatory submissions. But again, it's about that balancing that need for speed versus the predictability of timelines. I think if you're going to use a standard platform, it makes sense, as Brian points out in his paper, to use standard materials and consumables. If you change these consumables from the platform, then you need to raise things like new item specifications. You may need audits of, and this can all extend lead times. So sticking to platform materials and consumables helps save time. Getting into your first GMP batch, and that's balanced against the fact that you are somewhat constrained, and that you can only stick to the platform when manufacturing. And then finally on this slide, things like co-location of different parts of the manufacturing process, such as drug substance and drug product manufacture, can save a lot of white space in your timelines, because it avoids that shipping round of materials, but also it's much easier to schedule these different activities if they're performed all within the same site. But I think it's probably on the other side of the coin, and that need to produce very large amounts of material, which is scheduled for more of a shake-up in the next few years. What I tried to do in this slide is generally show a typical approach for a conventional program. So a company might start out with running a fairly small-scale bioreactor, maybe 200 liters, to generate material for those tox studies, and then scale up during phase 1, or just before phase 1, to scales such as the 2,000 liter scale, continue at that scale for phase 2, and then scale up to the largest scale that they intend to commercialize out of for phase 3 in their regulatory review. And what I tried to show with that gold arrow is just how long the investment decision into a new facility or an expanded facility requires, and how early it needs to be made within the development cycle. Now, I think what we've seen this year is that companies such as Regeneron and their manufacturing partner Roche have been able to scale up to these very large bioreactors very rapidly. And that doesn't particularly come as a surprise to me, because the reason this timeline is such is more linked to the duration of clinical trials, not how long it takes to scale up to the bioreactor and the large-scale facility. It's very possible, using the right scaled-down models, using the right control strategies, and having well-characterized equipment, it's very possible to scale up in a relatively short timeline to even the largest scales of manufacturing capacity these days. But what I wanted to suggest in second half of this presentation was just that there are possibly other alternatives to that large scale-up strategy, and that there may be benefits. So, for example, this slide is very similar to the last one. What it's trying to illustrate is the scale. But then, rather than going up to a very large scale, actually operating a facility with a large number of the same size bioreactors to avoid that scale. And these very high throughput fashion these days, and this avoids having to do that scale up, which has some risk and cost associated with it. And then I think very importantly, it allows the investment decision in new facilities or capacity to be delayed and made at a later date. And it's a very flexible approach, in that, given that there are uncertainties about demand for commercial demand for antibodies, it allows the capacity to be modulated very easily accordingly. What's driven the ability to kind of implement or execute, I suppose, a scale-out strategy are improvements in productivities of cell lines. So we can now get as much antibody out of a 2,000-liter bioreactor because of high titers than we were able to get out of a 5,000 or 6,000-liter bioreactor even just a few years ago. There's been advancements in maturity in single-use technology and in the use of single-use bioreactors that can be installed and operated in a very reliable fashion in large open plant facilities. And also, we increasingly see the adoption of the ballroom concept of facility design, so that multiple batches can be manufactured simultaneously within the facility in the closed process environment, without risking batch-to-batch product contaminations. And like I say, this is leading to help uncertainty in demand forecasts. And it also supports, as a colleague was saying to me, it really supports a decentralized strategy. So you could imagine, for example, one facility with 12 bioreactors in, firing off at a high throughput, but you could also located within regions, such as one in the States, one in Europe, and maybe one in Asia, which would have some benefits in terms of localization manufacturing, but also reduce some risks associated with supply chain security and ensuring that there's always a supply of drugs for the patients. I think the final manufacturing paradigm that I wanted to describe was really probably the most exciting of all, which is the implementation of continuous biomanufacturing for antibody production. And we've implemented a platform. It's more connected and integrated. So the final stage of the platform that we've implemented is actually operated in batch mode. But by applying kind of these heavily intensified processes, we are seeing that we can increase antibody output from a 500-liter single-use bioreactor by 5 to 10-fold. And it uses very much the same purification platform as we use in our fed batch processes. And that's important because it gives the potential to allow the the interchangeability of fed batch and continuous processes. So we may find that some customers will start off with a fed batch process and then transition to a continuous manufacturing process at a later stage. And again, this is allowing for demand, demand for large amounts of antibody to be met with a significantly smaller, much more agile manufacturing facility. So what I tried to do then is I tried to compare these three strategies for market supply by picking a kind of an arbitrary figure of 2 tons of antibody per year, and then trying to show kind of how many batches and bioreactors you might need in order to fulfill this demand based on that scenario. So if you look at the scale-up approach using multiple 20K bioreactors, by my calculations, a good cell line, you can fulfill kind of 2 tons demand of antibody with 25 to 30 runs a year. With the scale-out approach, you clearly need many, many more runs, so 160 to 170 runs per year. But again, there may be ways of doing that in localized facilities, or well, it's very possible to do this within a single facility with 10 to 12 bioreactors in. But I think what this really illustrates, this scenario really illustrates, is the effectiveness of continuous biomanufacturing to supply large amounts of antibody. In that 2 tons of antibody can actually be supplied from a facility operating in a continuous mode with just 4 500-litre single-use bioreactors, or 1 2,000-litre, which is really an incredible achievement by the industry, and will really help deliver antibody therapeutics to a very large number of patients moving forward. And again, the real benefit. So to summarize the part of the presentation, that therapeutic antibody pipelines are very robust. The pandemic's really increased the need for short development times and significant manufacturing capacity. And I've just tried to show some of the innovative approaches to development and manufacturing that the industry is adopting, and that hopefully we can leverage in the future to give the most efficient development of antibody candidates. With that, I've left my contact details on this slide, so if anyone wants to discuss any of these topics, please do get in touch. It would be great to do that. I wanted to thank Jan and the Antibody Society for giving me the opportunity to present today, and you, the audience, for your attention. And with that, I will hand back to Jan and the team for the Q&A session. Thank you.
Thanks very much, Nick. And I think Anjuli might be there. Anjuli, are you there?
I'm here. We do have a few questions. The first one is, and I am going to apologize if I do not pronounce these correctly, but the first one is REGN-CoV-2 equals 2.4 grams a vial. Does that mean that there is 1.2 grams Casirivine?
Don't worry about the mAb names.
Sorry, and 1.2 grams of
the other one.
Yeah.
That's okay. Sometimes I have to avoid those names too, because you have to parse them just right, and until you practice it, it mostly does not come out correctly. But the question is yes. So that's actually a relatively simple answer. The 2.4 gram dose is an equal amount of each of the 2 individual components. They are combined together in an IV bag of a total of 200 mils, and that's infused over the course of an hour. All of that information, I mean, it's not magic information that I was able to extract from anywhere esoteric. It's right on the FDA's website in the section that describes the emergency use authorization of the product. So there's the information is there not only for this mixture, but also for Lilly's molecules, for remdesivir. Not very many therapeutics have gotten EUAs yet, but the information is right there just on the FDA's website.
Great, thank you. Next question, do you know something about the Sorrento Therapeutics antibodies?
Yes, Sorrento is actually doing a lot of work. They have a number of antibody-based therapeutics or antibody-related therapeutics that they are examining, evaluating, developing. They have, right off the top of my head, I'm trying to remember them all because I think there were certainly at least 4, if not 5. 2 of them were antibodies, like real antibodies. One of them is DNA based, though, that encodes for an antibody. One of them, I believe, is a trap type molecule where it's ACE, so it's the target of the virus, ACE2, fused to an FC, I believe. They have a variety of approaches that they are taking. I think that company has the largest number of distinct molecules that I'm aware of. All of them, though, are in early stage development. So they're either still in preclinical development or maybe at most 2 have gotten to early stage clinical studies. So I didn't talk about them in the presentation because I didn't talk about most of the ones that are in clinical studies. I limited myself to the ones that are in late stage clinical studies. If you go to the biologics tracker page on the Antibody Society's website, there are something like 19 listed in that table, including the Sorrento molecules that are in early stage development. And again, if you want to look at them all, the Excel spreadsheet is right there. There's over 200 molecules listed. So if you're really curious about all the information that I've got, I have made it available to you. Just go to the Antibody Society's website and find it.
Great, thank you. Next question, do you know if an MD has discretion, like off-label uses of approved drugs, to give either of the Lilly or Regeneron antibodies to a hospitalized patient despite what the EUA says? Thanks so much for the great presentation.
Well, you're welcome. That's a very good question because typically physicians do have a lot of leeway when products are approved. They can do this off-label use thing. I'm not sure myself that that's actually something they can get away with under an EUA. An EUA is not an approval. There are very distinct restrictions associated with emergency use authorization of these products. So I would say, if pressed on the point, my inclination would be to say no, they cannot do that. Mostly because it is explicitly excluded as a use. And since that's done, what I don't know is if there's some specific mechanism. I don't really know, I don't believe it's been made public, the plan for who is going to get this, when they're going to get it, where they're going to be doing these infusions, because they are an infused product. I didn't mention also about the formulations, but the IM formulations are being investigated by AstraZeneca, certainly, and subcutaneous administrations are being investigated by a number of organizations, I believe also Regeneron. So that would make it a lot more convenient for patients if it could be IM or subcutaneous administration. Having to go somewhere to get an infusion is definitely going to be inconvenient. Well, unless you're already in the hospital, then it's not so inconvenient. But I believe right now, until the late stage studies of the hospitalized patients are done and there's positive results from that kind of a study, I believe it's the use in hospitalized patients is excluded.
Okay, thank you. Next question, does the number of mAbs, I think that's how you say that, on slide 19 include next generation formats such as bispecific antibodies?
I don't exactly remember which slide was 19, but what I can tell you is that the slide that shows the graphs, and it shows something like 90 programs in preclinical or discovery, that does include some nanobodies. It does include, you were on, this is slide 19. Never mind about what I was just saying. In this case, it does not. But in fact, I'm not sure it would have changed these numbers dramatically, considering there's only 19 in clinical studies at all. So what I would go back to is the slide that does have the programs. It looks very similar to this, but it has a bar showing something like 90 preclinical and discovery programs specifically for, keep going back, specifically for the COVID-19 biologics. And that definitely, yeah, there's the one. That definitely includes the nanobodies, the sort of alternative things. There's some IgYs in there. There's some really interesting and offbeat stuff in that discovery and preclinical space. And again, every single scrap of information I have about these, at least the summary information, is available in an Excel file that you can download from the COVID-19 biologics tracker page. And I do keep updating that, but I only post an update about once a month, just so you know.
Okay, thank you. We did have another question come in. Several escape mutants have recently been identified for both the Lilly AB and the Regeneron cocktail. Are you aware of any AB therapeutics in development actively screening for ABs targeting escape-resistant epitomes?
Yeah, I'm not aware of that, so I don't believe that I can comment on that point. But of course, this is something that is of concern, that you've designed your antibody to bind to a specific epitope, and if the virus has been mutating such that it does not bind anymore to the escape mutant, then that is definitely problematic. But I am not aware that anybody is specifically addressing that problem already. So I think that's relatively new information. So I actually have a question for Nick, and considering the amount of material that might possibly be needed for some of these products, how do you imagine that need for the production capacity is going to affect production of these other molecules that are already on the market or in clinical development now? Is there a crunch on production capacity, I guess is the question.
I think, yeah, that's a really good question, Jan. I think not just for manufacturing, but also for how quickly these other molecules can be developed. I think that you can imagine that there is a finite amount of both development and actual then production capacity in the world. So I think that's why we need to look for these innovative approaches to prevent that becoming an issue, because I think really you could see that will be a challenge, and that these anti-SARS-CoV-2 antibodies could kind of
I don't see any more questions here for now.
Okay. I think we have maybe one more minute, and I think this is an easy question, because the answer should basically, I think it would be just yes. And Nick, do you think, because so much has been learned that so many parts of the process can actually go faster than we thought, is this likely to affect antibody therapeutics development in the future?
I certainly do. And I think that there is possibly an element here, though, that we would want to move faster during a pandemic and have moved faster. So whether all of these ideas for accelerating timelines will continue to be implemented after the pandemic or for non-pandemic mAbs, I'm not quite sure, because it probably leads to a greater risk profile on those projects. But I think we will take some of those ideas forward into antibody development in the future.
Well, with that, I believe we've reached the end of the hour. I do thank you very much for coming and listening to us today, and I hope you go to the Antibody Society's website and take advantage of the resources that we've made available for you there. Thank you.
Thank you.